So next we are going to move to the panel discussion. The moderator of the panel is Dr. David Weiner. He is in the producer room, but I'm waiting for him to turn on his this monitor. While I'm waiting for him to turn on the monitor, I can give a little bit of background of this panel. We have three really esteemed presenters here and panelists. Actually, we are going to ask a very simple question because this is clearly related to the final phase 3 test of efficacy. Most emerging infectious disease will come very potently, but then they will disappear very quickly. So the problem is how are we going to have a proper population to do a test? Of course, if you have a high risk group, the efficacy will be much easier to be demonstrated. Okay. So that's why it has been proposed. Also, was the way practiced in the history of vaccinology. That is, you can actually challenge the human volunteer with the same virus. Okay, now I see my colleague David Weiner is there. So I will yield my video to him, and he will continue and with the three panelists. So Dave, now it's your turn.
Thank you, Shan, and thank you, ISV, for such a wonderful Congress and to all the exceptional presentations today. It's really an amazing accomplishment by all the speakers to advance so much in such a quick period of time. There was so much thought. So along those lines, one of the topics that's been discussed very significantly recently is with such a pandemic, should we consider an unusual strategy that's the advancing really a live human challenge model? And such a model, of course, has significant conceptual advantages, but it also has some issues that still need to be addressed. And we have assembled ISV has assembled an exceptional panel of individuals to discuss this topic. In order of their speaking will be Dr. Stanley Plotkin, who is an emeritus professor of the University of Pennsylvania and adjunct professor at Johns Hopkins, and a former member of the Wistar Institute. He's a world authority for vaccine development, having developed the rubella vaccine and co-developed the pentavalent rotavirus vaccine, as well as contributed to development of 7 additional vaccines, translational development. And he has received many awards and honors, and serves on many committees that are essentially important, including chairman of the Infectious Disease Committee. And I'll stop there in the interest of time. And
so Dave, sorry interrupting. We have the slides for actually Peter Openshaw first. So if you don't mind, introduce him. So the order will be Peter first, and then Stanley Pearlman and Stanley Plotkin, if that's okay with you, because our slides are set up that way.
Right, but isn't Stanley starting as the reasons for? And so it would be.
Well, let me ask Ben. Ben, can you bring Dr. Stanley Plotkin's slides first? If David has decided, that's fine. Okay, so Peter, I hope you don't mind. You will have the counter punch. We'll save you to the last. Okay.
Well, I can ask Peter if he was going to go first. I had the wrong information. So Peter.
But that's okay. Our slides is up. So Stanley, Dr. Plotkin, can you start? I can see you are ready. You have a full gear there. Okay, let's go with you first. How Ben put his slides up. Oh, you are mute. Stanley, you need to unmute. You see a little microphone on your screen. You just click that one, that should do it. you can see there's a bar. If you move your mouse, you should see a bar. There's a microphone.
Yes, okay.
Now we can see you. Thank you. Go ahead.
Okay, right. So thank you, David. And so this is under the heading of extraordinary times require extraordinary measures. And I have just a few slides, and most of what I want to say will be on the last slide. But the next one, please. So as you all know, the standard vaccine development starts with phase 1, a small number of people, progresses to phase 2 with a somewhat larger number, and then the phase 3 trial requires a lot of people and usually a fair amount of time in order to collect illnesses in the vaccinated and the placebo groups. But what I wanted to stress also in this slide is that what you want, paradoxically, is disease in the placebo controls. And in the case of COVID-19, one would anticipate not only illness, but deaths as well in some of the placebo controls. Next slide. Well, you all know, when you've just heard about the many different vaccines being developed and tried against the SARS-2 virus. And so I don't think I need to read this slide, but just to emphasize that there are many different platforms being used, many different companies involved, so many different vaccines that need to be tested and shown to be effective. Next slide. This slide simply emphasizes the many different vaccines in development, each one of which will require demonstration of efficacy. So this is quite different than prior vaccine development, where there were only a few candidates and where it was possible to show efficacy because there was little or no competition. Next. So this is my last slide, and let me go through this in detail. So what we have proposed, and when I say we're not the only ones who have proposed this, but what has been proposed is to use human challenge as a way of accelerating vaccine development and use. So what are the possible advantages? Well, fewer people, many fewer people are needed for human challenge studies. And one would, of course, choose the group that has the least likelihood of serious disease, which would be the 18 to 25-year-olds. And with such a group, one could determine the infectious dose of the virus, because one would start with a low dose in order to, what shall I say, to challenge with the smallest possible dose. And this would be of some biological interest also, to know what the infectious dose is. One could, of course, increase the dose if there were failure to infect. I should stipulate that this means that there would have to be some seronegative individuals in these trials. One could not solely test vaccinees, because one would need to verify that they are indeed receiving an infectious dose. Another advantage is that it will be possible to compare vaccines. How are we going to do phase 3 trials on the 100-odd vaccines? Well, probably that will never happen. But even if there are only 10 or 12, that's quite a lot of of phase 3 trials that one would have to do. Another point is the possibility of determining correlates of protection, because in this kind of study, you can do all kinds of immune measurements, antibodies, cellular responses. You all know the myriad immune responses that we now can measure. And if a vaccine is shown to be effective in the human challenges, one could obtain from the regulators emergency use authorization, which would mean in high-risk people like healthcare workers. Now, the last point is debatable in a sense, and that is that this could be faster than phase 1 to 3. I say this taking into account that one has to produce the virus, and one has to make sure that it's clean, but this can be done now with next generation's sequencing. So the claim that it's faster will depend on circumstances. Fortunately, the NIH has agreed to produce a potential challenge virus. Whether they will agree to use it is now uncertain, but at least there will be a challenge virus of SARS-2. What are the disadvantages? Well, obviously, danger to the volunteers. I should mention that it's quite interesting that there are literally thousands of people who are willing to volunteer. Now, obviously, they have to be selected. They have to go through psychological testing and all that, but there's no lack of volunteers. Another problem is rescue therapy. It is true that we don't yet have an absolutely clear rescue therapy, but one hopes that in the near future, there will be antivirals and immune serum monoclonal antibodies that could be used. And lastly, challenge studies will not give you sufficient safety data. And here is my last point, that the challenge trials do not eliminate phase 3 safety data. You're still going to have to test in large numbers to obtain a license, but at least you would know that the vaccine works and that you would be able to eventually prevent the collapse of society due to the economic effects and the human effects, which are currently devastating. And I'll stop there.
Okay, thank you, Dave. I think the next one, based on our slides right now, is Dr. Stanley Perlman. Do you mind to introduce him?
Thank you, Shan. Thank you, Stanley. Professor Stanley Perlman is a world-leading authority in virology and immunopathogenesis of viral infections, specifically coronaviruses. He's advanced seminal research efforts focused on pathogenesis, animal model development for SARS, MERS, COVID-19. He's received many, many awards and honors and served on important distinguished advisory panels. In the interest of time, I have to just bow in his general direction and now allow Stanley, Professor Perlman, to present. Thank you so much for being here today.
Okay, thank you, David, and thanks, Shan, for inviting me. And it's a pleasure and an honor to follow Stanley Plotkin, the other Stanley P in this group. So, so can I have the first slide or the next slide? So I come from this from a little different perspective because I've been thinking about coronaviruses for a long, long time. And there's several things that just led me to try to answer this question from that knowledge. So the kinds of things that are that I think we need to know, and this has been covered in many of the talks already, is what is actually a protective immune response, and does natural infection induce a protective immune response, and what is the longevity of the response, and do the various vaccine formulations induce a protective immune response? And again, this is what I'm trying to do here is just tell you the way I'm approaching this problem. So could I have the next slide, please? And so how could experimental human vaccine trials help? So they could allow measurement of antibody and T-cell responses under controlled circumstances. They could allow assessment of protection under experimental rechallenge. They could assess vaccine safety and efficacy in head-to-head trials of different vaccine formulations, and could allow assessment of effects of vaccine and prior infection on clinical disease and shedding. And this may be one of the most important points, because shedding, as was mentioned in some of the earlier talks, is something that we don't really have a great handle on. Even the volunteer studies from many years ago didn't really quantify shedding. They just showed that in some cases, as I'll mention very briefly, shedding occurred. So could I have the next slide, please? So what are the risks that I saw with experimental human vaccine trials? So one is the development of pneumonia, but this seems to me to be less likely or unlikely in the younger populations that are being considered for these kinds of trials, because this is the population that's 15 to 25 years old, or about that range. The second point I'm a little more worried about, which is the development of this vasculitis hyperinflammatory disease. I don't know that anyone understands this well. We think it's a dysfunction of endothelial cells, but there's little evidence for endothelial cells being directly infected. So it occurs sporadically. It doesn't occur in large numbers of people, and it could be genetically based, but it's really not understood, and it's not predictable. And I don't know a way to screen volunteers to see if they actually would be susceptible to this outcome. And of course, there's no proven therapy, particularly for the hyperinflammatory disease, if infection becomes severe. And I should say this disease, this Kawasaki's-like disease, was initially reported in children, younger children, but it's seen in this age group as well. And I don't know whether this is a continuum of the thrombosis vasculitis syndrome that's seen in older people, which is also not well understood. And it may be possible to use remdesivir for inhibiting viral replication if experimentally infected volunteers seem to be becoming more ill. This may not work, though, on that hyperinflammatory illness. Could I have the next slide, please? So there's several other things to consider, and this is just a list of some of them. Are there strains of virus that differ in pathogenicity? There's definitely different strains of virus, but not clear that they differ in pathogenicity. That's under study and controversial. What dose of virus should be used? How long after vaccination will a protective immune response remain? When should rechallenge occur? Would you want to rechallenge people 4 to 8 or 12 weeks after vaccination when it's more likely to succeed, or do you want to wait? At the other point that was already made was the longer you wait, the longer the results will take to come in. And then the other point is that there's so many people now who have been infected, even in this country, it's probably at the 1 to 10% level. We can learn a lot from experimenting infecting COVID survivors who have well characterized and high antibody and T-cell response. So this will tell us information about the immune responses after natural infection and how much protection there is. Could I have the next slide, please? And so, in some of these, I think some of these questions have been answered from our previous studies of common cold coronaviruses, MERS coronavirus, and SARS coronavirus. We know that antibody responses generally develop after all of these entities, and we know that the milder the infection, the more likely the antibody responses to be low and transient. In MERS, we saw patients who never developed antibody responses, even though they were infected. The more severe the infection, the more likely that the response is long-lasting. And this is, I think, the next point is key. Immune responses may prevent clinical disease, but not shedding. Shedding is so important to quantify, not only just to demonstrate plus or minus, but to quantify. In the volunteer studies that were done with common cold coronaviruses, there was evidence for protection against the cold, but not against shedding. But I don't remember that the amount of shedding was ever quantified in those experiments. And T-cell responses, which have been discussed, may exhibit more longevity. And I think SARS-CoV is very likely, we don't know for sure, very likely to follow these same response patterns. So can I have the next slide? So in summary, will human vaccination trials be useful? So I think infection of COVID-19 survivors would provide information about rechallenge without needing to expose naive volunteers to infection. And human vaccine trials would be most useful, should be performed to evaluate relative vaccine efficacy. As Stanley pointed out, we're never going to be able to do head-to-head on all of these vaccines, but at least if we can do some, we can get information about which ones really seem to induce the most protective immune responses and how long they will last. And volunteer studies may be useful for identifying biosafety issues in the short term. This is going to be very short term, and so we'll probably not identify maybe the most important ones. The measles problems with the measles vaccine from the 1960s took a few years to actually be fully apparent. And then the other thing I think is that in the US, there's so much vaccine skepticism. I think if there's good data from these human volunteer studies that the vaccines are working and are safe, that'll do a good job in increasing the percentage of people who are willing to be vaccinated from the 49% that I read most recently to a number that's more consistent with generating herd immunity. So for me, the answer is a qualified yes. And with that, I'll stop.
Can't hear you, Shan. Hello, can people hear me?
Yes.
Okay, go ahead. Sorry, you go ahead to the last speaker introduction. Yeah.
Right, so it's an honor to introduce Professor Peter Openshaw, who's Professor of Experimental Medicine at Imperial College and honorary physician, Department of Respiratory Medicine at St. Mary's campus at the Imperial College. He is a renowned investigator on the immunology of the lung, viral lung disease, vaccination, immunopathogenesis of viral disease. He has many honors, including that he's among the first 100 people selected as a fellow of the Academy of Medical Sciences. He served on many international committees and received many prestigious awards. Again, in the interest of time, Professor Openshaw, please welcome, and we're looking forward to your talk.
Well, thank you very much indeed. It's an enormous honor to be on the same podium as Stanley Perlman and Stanley Plotkin, even if only virtually. So I think it's been a fantastic meeting so far. It's a shame we can't all meet each other and go and have a drink afterwards to talk about all we've learned. I'm certainly feeling so much more positive and enthusiastic that we will have vaccines and we'll be able to get out of this terrible lockdown that we're all placed in, many of us are placed in at the moment. So the first point I wanted to make was that actually experimental infection of human volunteers is, if anything, on the ascendant. So you would have thought that it might have been superseded by other techniques. But if we look at this first graphic from Mette Rosenberg's lovely review in the Lancet Infectious Diseases a few years ago, you can see that actually the number of vaccine challenge studies has been climbing. So they're by no means in recession. They've been done for a very large number of pathogens. And you can see those listed on the right. So I think the list of pathogens that it's been thought ethical to infect with is quite extensive and includes some pathogens for which there is very little by way of mitigating therapy. If we go to the next slide, I think if anyone had told me back in 2009 that we would be infecting volunteers with this pandemic strain of influenza, the H1N1 pandemic swine flu strain, I would have just not believed it. But in fact, that's what we've been doing. So Chris Chu has been performing these experiments in my group for some years now. And we get reliable infection. I think it's remarkable the amount of information you can glean by these studies in terms of being able to investigate the local mucosal response and also the systemic response. It's hugely revealing in terms of the detailed immunology that you can get. If you look at this lower graphic on RSV, I think one of the most remarkable things is the lag phase between inoculation and the development of symptoms and viral replication. And we're fascinated by that sort of interval during which there is occult negotiation going on between the pathogen and the host. What exactly is happening during that critical phase where a decision is being made, whether one or the other becomes triumphant? I think the immunology that you can glean by these sort of experimental studies is fascinating. We've got a consortium that I'm proud to have been able to lead for the last few years called HICVAC, which is Human Infection Challenge to Promote Vaccination. This is funded by the Medical Research Council. in the UK, and it's a truly international consortium to try to promote the use of human infection challenge, particularly for the benefit of pathogens of global importance. So next slide is my last, and this just sums up my current thoughts about whether it is okay to go ahead and do SARS-CoV-2 challenge in people with young people with no comorbidities. And just if I can first direct you to the bottom half of the slide, this is an amazing consortium that was set up in the UK, of which I'm one of the co-leads, and we've actually managed to recruit 55,000 COVID positive people in hospitals so far, which is about over 45% of all of those who've been admitted to hospital in the UK. So this gives us a huge database of information. Many of these patients we have biological samples on, and some serial biological samples up to day 28. But just in terms of whether it's okay to challenge young people, we have seen young people with no comorbidity who have actually had a fatal outcome. So I'm very hesitant to recommend that this should be added to the list of the challenge studies that it's okay to perform. I think we need very careful selection. We don't yet know whether there's some genetic propensity that leads to the deaths in these unusual few. It has to be done under very tightly regulated conditions, and we need to be able to bring in antivirals to terminate the disease or to mitigate the impact. And I think day by day we're learning more about how that can be done. So I think, you know, the inherent limitations to doing these human challenge studies, you know, they don't tell you necessarily what would happen in high-risk populations. It's not easy to predict so far the few who might get more severe disease. And we still don't know about the long-term sequelae. You know, we're learning more and more about this virus with a really nasty sting in its tail. And at the moment, I'm a little cautious. I think we should be moving forward to do these studies, but by the time we've actually got all the approvals in place, my fear is that we'll have been overtaken by the field studies, some of which you heard descriptions of earlier today. So that's my current feeling. I think we should be moving forward, but I'm very cautious. Thank you.
Well, thank you very much. We've heard from all three speakers, and that was wonderful for setting the table. So I see there are some questions coming in, so I will start with those. So first, for anyone in the group, I guess maybe Stanley leading off, what does a challenge in a healthy 18, 25-year-old mean, and the immune responses in the high-risk groups, including older people? How would you bridge response or safety to these groups?
Well, so do you hear me?
Yes.
Yeah. Okay. So that of course is quite a reasonable question, and my answer is this: that if you identify the immune responses that are protective, whether they be antibody mediated or cellular mediated, then you can look in the elderly receiving the same vaccine to see whether those immune responses are generated. We all know that the elderly do not mount immune responses as well as the young, and I think it's likely that this will be true for SARS-2 as well. Nevertheless, if we are going to find a vaccine that is useful in the elderly, we need to know what are the immune responses that will protect them. And I think the challenge situation could identify those immune responses. By the way, just let me comment that I think both of my colleagues really were quite fair in their evaluation of this subject.
So another question, I'll go to the second Stanley. Determining challenge dose can affect challenge outcome. We heard earlier in the session that the MERS, I believe it was, challenge used a very high dose and so may not be completely reflective. And I guess I'll add to this. So if we need to do challenges in small number of people, we will be likely trying to infect all of them, which is likely not in the fields. Dr. Pearlman, so how do you view this kind of challenge in such a setting?
Yeah, so I think that the first thing you have to do is establish an infectious dose. So that's the best answer I can think of. So you do a range of dosages, and you can measure both virus loads in the nasopharynx afterwards. You can monitor clinical disease. You can monitor immune responses. And I think that kind of information has to precede any kind of further studies you do. The other thing is, I just wanted to add something to Stanley's comments on the implications for immune responses in aging. One of the things that we've noticed mostly from our experimental studies is that in addition to immune responses, and again, it's experimentally infected animals, in addition to the immune responses per se, there also seems to be differences in the lung milieu that may impact, even with a good immune response, how the host responds to those parameters in eliminating the virus. So that may be another feature that we have to think about, because it may not just be strictly a numerical correlation. There may be additional factors, but we still need to know those numerical numbers, those numerical factors on what constitutes an immune protection.
So the next question, I guess, is if we were to have a negative outcome in one of these challenges in the control group, how would that be viewed? And is that an issue we have to consider? So, I guess I'll start with you.
Sorry, it cut out a moment. Is that for me?
Yeah.
I think.
Well, you can start, but then
Yeah.
I think you should let all of you guys.
Absolutely. I mean, clearly, any adverse outcome that is clinically significant could be detrimental to the whole field of human challenge. I mean, there was an incident in days gone by when a volunteer was infected with influenza and developed a cardiac abnormality. Now, whether that was pre-existent, we don't know, but that really held things back in the US. I think in the UK, we're a little bit more tolerant. Our population is pretty amenable to being experimented on for some various reasons. And I remember my own son, when he was aged about 7, asking me what I did. And when I explained, he went quiet for a while and then said, "Dad, if you want to experiment on me, you can." So that's sort of representative of the willingness of the UK population to participate. But I mean, clearly, adverse events would be potentially disastrous to the field. Yeah.
So another question that really was mentioned, I think, in Perlman's talk as well, is that there's recent reports that, for example, Michelle Nutson's weed, that there's very low titer or almost no titer in some percentage of people who have relatively mild disease, that on the U.S. aircraft carrier, that was a report in MMR reports that a significant percentage did not develop antibody responses and yet had mild disease. And now there's another report from China as well as others emerging. So how do we figure that into these type of trial designs?
Yeah, can I?
Sure.
Sorry.
Sure.
I mean, we don't know whether the things we're measuring are actually going to be particularly predictive of protection. And the assays are evolving over time. I think some of the initial antibody assays that were developed were not particularly sensitive and missed a proportion of those. But I think we really need to see what the correlation is between the things that we measure and the outcome. We just don't know quite enough about that yet.
I think I heard Dr. Plotkin also wanted to address that question.
Well, I think it boils down to what sort of immune responses can be generated by the intranasal challenge. And I also have read that some people do not respond, presumably because their viral load is not high. All that I can say is that it is a factor that we will have to work with. We don't want to induce serious disease, and so it may be troublesome. That is the lack of an immune response, if that's the case. Nevertheless, on the other hand, you could say that might be a guarantee of safety, and that factor, I mean, and that we will still need to look for immune responses if there are any. So it may be a problem, but on the other hand, we will never know until we try it.
Yeah, and I just want to add one other thing, which is even in the studies, the MERS studies, which I know best, where there was very low immune responses, we don't really know that even if people had very low responses, if they weren't protected from pneumonia. Because I think that you think of vaccination as two parts. One is protecting people from pneumonia, so the individual is infected. And as I tried to mention also, and others have as well, the societal one is actually diminishing shedding, because if we can protect everyone from pneumonia, that's great. But if there's still 80% who are susceptible to shedding, then we want to eliminate that. And that's really one of the key things that we don't know whether these low titers will protect against that. From the common cold ones, as I mentioned, it seems not, but we don't really know the level. But for me, that's the really important parameter that we want to get out of all of this.
So, Dave, I think due to time, we have to stop here for the panel. We never expected the panel discussion will work so well the first time we do a virtual conference. So, Dave, according to our program, you need to give a summary of this panel discussion.
So I guess, first of all, I'd like to thank the three outstanding panelists, Dr. Plotkin, Dr. Perlman, and Dr. Openshaw, who really gave us a lot in just a very small period of time. I think we heard there are positives and issues that still need to be addressed, and we also learned about where they can go, and I think that this is a great foundation for additional discussion and moving forward and figuring out the value and how to place such studies in the overall context of field development, especially in a very, very unusual time where, as Professor Openshaw mentioned and Dr. Plotkin mentioned, we're moving so fast in efficacy through field trials. So I think this is a really important topic, and a very exciting and relevant. And so thank you again for your discussions and enlightening us.
Thank you, Dave. That was a very good summary. Now we have to move on to the last item of our program. Let me introduce Dr. Margaret Liu, who was a past president of our ISV, give a summary closing remark of today's program, also introduce the next Congress. Please.
Thank you
Margaret.
very much. Thank you, Shan. First of all, I want to thank again all of our amazing speakers. I think that we have had a really good first session that has brought us up to date and given us all the important background about many issues. So just to remind you all that we will be then having, this is just the first in a series. And so we invite you to join us on July 21st on a Tuesday, and we will send out an announcement about the detailed time. We're trying to find a way that can include as many people around the world as possible. So we apologize for those of you for whom it was very late or for whom it was very early. I was up at 4:30 this morning, so I know what it feels like a little bit. So I'd also like, though, to thank the team that was behind this in the background, who did a lot of work to make sure that this could run on a virtual platform. And that, of course, especially includes Professor Shan, who has done a lot of legwork to make this happen. And then, of course, Professor Linda Klavinskas. Now, you all got an email that announced that one of the aspects of this symposia is that we send you a poll question for you to answer. And so before we had this panel discussion, we had the vote about whether or not you would feel comfortable with doing the challenge trials, infection trials. And I just wanted to announce the vote, which was that 75% were in favor of doing them and 25% were against doing them. So what we'd like to do is we will be sending out another email to you, and we'd like you to vote after you've heard our panel discussion, just to see what the vote is after you've heard the panel discussion. I also did want to thank all the audience for your participation. We've been very pleased with the ability to see many of the questions that you asked. And while for time reasons we weren't able to address all of them, we do hope that when you attend the next time, you continue to submit. We will make an effort to actually forward the questions that have been submitted to our speakers today, and we'll try to explore if there's some way that we can provide answers, even though we couldn't address all of the questions here. So thank you again for your attendance. Thank you again to all our speakers and the organizers, and we'll see you approximately a month from now at our second ISV Virtual Congress.
Thank you very much. This will conclude the first ISV Congress. Thank everyone. Goodbye, good night for someone stay very late. It's time to sleep, or good morning, someone is up all the time. And we will see you in about 1 month. Thank you.